Carl Correns and Eric Benchmark, who claim to have reached similar conclusions in their own research. So specifically, in 1889, Hugo de Vries published his book, Intracellular Pangenesis. And pan, already we've talked about it being all or whole. Pangenesis. Genesis simply means birth or beginning, right? Pangenesis. Hugo de Vries, his book, Intracellular Pangenesis, in which he postulated that different characters have individual hereditary carriers, and that inheritance of specific traits in organisms comes in particles. De Vries called these units pangenes in German, after Darwin's 1866 pangenesis theory. So 16 years later, that is in 1905, well, Johansen introduced the term gene, and William Bateson that of genetics, while Edward Strasburger, amongst others, still use the term pangene for the fundamental physical and functional unit of heredity. So you see that it has undergone several transformations till what we are using now. So the discovery of DNA, advancement made in the attempt to understand genes and inheritance, continued throughout the 20th century. Deoxyribonucleic acid was shown to be the molecular repository of genetic information by experiments in the 1940s as well as in the 1950s. So the structure of DNA was studied by Rosalind Franklin and Maurice Wilkins using X-ray crystallography, which led James Watson and Francis Crick to publish a model of the double-stranded helix structure, double-stranded DNA molecule, whose paired nucleotide bases indicated a compelling hypothesis for the mechanism of genetic replication. So each particular component base had its corresponding base that it had to pair with. So in the early 1950s, the prevailing view was that the genes in a chromosome acted like discrete entities, indivisible by recombination and arranged like beads in a string. So the experiments of Benza using mutants defective in the R2 region of bacteriophage, T4, that is, it was experimented between 1955 and 1959. It showed that individual genes have a simple linear structure and are likely to be equivalent to a linear section of a DNA. So collectively, this body of research established the central dogma of molecular biology, which states that proteins are translated from ribonucleic acid, which is transcribed from DNA. So it is RNA originates from DNA. That is why we say that proteins are translated from RNA and transcribed from what? DNA. So this dogma has since been shown to have exceptions, such as reverse transcription in retroviruses. So the modern study of genetics at the level of DNA is known as molecular genetics per discourse. So in 1972, Walter Pius and his team at the University of Ghent were the first to determine the sequence of a gene in 1972. Walter Pius and his team at the Ghent University were the first to determine the sequence of a gene for bacteriophage, MS2 code protein. MS2 code protein. So the subsequent development of chain termination. It's a long chain. Then you terminate it. You have to do away with certain parts. Chain termination DNA sequencing in 1977 by Frederick Sanger improve the efficiency of sequencing, and turn it into a routine laboratory tool. So an automated version of the Sanger method was used in early phases of the Human Genome Project. The theories developed in the 1930s and 1940s to integrate Mendelian genetics with Darwinian evolution called the modern synthesis, a term introduced by Julian Huxley. Evolutionary biologists subsequently refined this concept, such as George C. Williams' gene-centric view of evolution. So he proposed an evolutionary concept of the gene as a unit of natural selection, with the definition that which segregates and recombines with appreciable frequency. So in this view, the molecular gene transcribes as a unit, and the evolutionary gene inherits as a unit. Stated ideas emphasizing the centrality of genes in evolution were popularized by Richard Dawkins. So, in review, the idea of the existence of discrete inheritable units was first suggested by Gregor Mendel. And he explained his result in terms of discrete inherited units that give rise to observable physical characteristics, although he did not use the term genes. Initially, he was just experimenting, but he didn't go about using the genetic terms just as, or the glossary we use in genetics as we are using today. Mendel was also the first to demonstrate independent assortment, the distinction between dominant and recessive traits or characters, the distinction between a true zygote and homozygote, and the phenomenon of discontinuous inheritance. So his work went largely unnoticed after its first publication in 1866, but was rediscovered in the 19th century by Hugo de Vries, Karl Correns, and Eric von Schmack, who reached similar conclusions in their own research. If you remember last week, we discussed about these people. One was from Austria, the other from Germany, and Holland, in that order. So specifically in 1889, Hugo de Vries published his book Intracellular Pangenesis, in which he postulated that different characters have individual hereditary carriers, and that inheritance of specific traits in organisms comes in particles. So de Vries called these units pangenes. Pangenes. Pan, meaning what? Oh. And genesis, or gene, meaning what? Beginning, after Darwin's 1868 pangenesis theory. So advancement made in the attempt to understand genes and inheritance continued throughout the 20th century. Deoxyribonucleic acid was shown to be the molecular repository of genetic information by experiments in the 1940s to 1950s. So the structure of DNA was studied by Rosalind and Morris Wilkins using X-ray crystallography, which led James Watson and Francis Crick to establish a model of the double-stranded DNA helix structure or molecule whose paired nucleotide bases indicated a compelling hypothesis for the mechanism of genetic replication. So each particular base pairing had its corresponding nitrogenous base to be paired with, so that it continues its enzymatic processes with regards to protein synthesis. So on that note, bring Unit 2, Session 1 to a close. Now let's move on to Unit 2, Session 2. In Unit 2, Session 2, we'll be looking at experiments to prove that DNA is the genetic material. Experiments to prove that DNA is a genetic material. And before this preamble, of late, there are a lot of paternity issues, and you realize people going for DNA analysis to verify paternity of most of the kids that are being born nowadays. So by the end of this session, you should be able to understand some DNA transformation experiment, and again, explain the DNA transformation experiments that also confirm the DNA as genetic material, Sorry for that. Okay, sorry for that. So you should be able to explain the DNA transformation experiments that also confirms the DNA as a genetic material. Now let's move on. So scientists first thought that proteins that are found in chromosomes along with DNA will turn out to be the sought after genetic material. Proteins were known to have diverse amino acid sequences, while DNA was thought to be a boring, repetitive polymer, due in part to an incorrect but popular model of its structure and composition. Today, we know that DNA is not actually repetitive and can carry large amounts of information. It's discussed further in the article on discovery of DNA structure. But how did scientists first come to realize that boring DNA might actually be the genetic material? Friedrich Griffith, bacterial transformation. In 1928, British bacteriologist Frederick Griffith conducted a series of experiments using Streptococcus pneumoniae, bacteria, and mice. Griffith wasn't trying to identify the genetic material, but rather trying to develop a vaccine against pneumonia. So in his experiment, he used two related strains of bacteria known as R and S. These are strains of bacteria because he was trying to develop the vaccine to combat the dreadful pneumonia. So the R strain, when grown in a petri dish in the lab, the R bacteria formed colonies or clumps of related bacteria that had well-defined edges and a rough appearance, hence the abbreviation R. So the R bacteria were non-virulent, meaning that they did not cause sickness when injected into a mouse. Now, let's look at the S. The S strain, on the other hand, S bacteria formed colonies that were rounded and smooth, hence the abbreviation S. The smooth appearance was due to a polysaccharide or a sugar-based coat produced by the bacteria. This coat protected the S bacteria from the mouse immune system, making them virulent, capable of causing disease. So mice injected with live S bacteria developed pneumonia and died. Frederick Griffith, his bacterial transformation. So as part of his experiment, he tried injecting mice with heat-killed S bacteria. The bacteria was subjected to heat. That is, S bacteria that had been heated to high temperatures, causing the cells to die. So unsurprisingly, the heat-killed S bacteria did not cause disease in mice because in the first place, the strain has been what? subjected to intense heat and that all the bacteria had died. So the experiment took an unexpected turn. However, when harmless R bacteria were combined with harmless heat-killed S bacteria and injected into a mouse, not only did the mouse develop pneumonia and die, but when Griffith took a blood sample from the dead mouse, he found that it contained living S bacteria. So the rough strain.
Hello, Doc.
Yes.
Hello, Doc.
I'm here.
Please, does it mean that the R bacteria wakes the S bacteria up?
Come again.
The R bacteria, which has been found in the dead mouse, does it wake up the dead one, which is the S bacteria?
More or so. Okay. It ignited it to, for you. Let's look at the various implications that was raised. The rough strain, non-pathogenic.
Hello,
right?
sir.
And this strain, yes.
Sir, please, is there any diagram to illustrate this?
You will definitely have it. So please keep your calm. There are lots of diagrams signifying this. Okay.
Okay, sir. Thank you.
All right. So keep your calm. We are moving step by step. Is that right?
Yes, sir.
So the rough strain, the R strain, okay, when this strain is injected into the mouse lives. A smooth strain, the pathogenic, when this strain is injected into the mouse gets pneumonia and dies. Heat-killed smooth strain, the S strain. When heat-killed smooth cells are injected into the mouse lives because the strain is what? Denatured. It is dead. Raw strain and heat-killed smooth strain. When these two types of cells are injected into a mouse as a mixture, the mouse gets pneumonia and dies. But if only R, which is the raw strain, it is not pathogenic. When it is injected into the mouse lives. The combination of the two, that means there is what? A heterozygosity occurring. So that will make the mouse develop what? Pneumonia, die. So Griffith concluded that the R strain bacteria must have taken up what he called transforming principle from the heat-killed S bacteria, which allowed them to transform into smooth-coated bacteria and become what? Virulent. So you see the conclusion. The R, when it is injected into the mice, it didn't die. But when S is injected into the mice, it developed pneumonia and died. When heat is applied to the S, it denatured the S. It killed all the bacterial cells in the S. And when it was injected into the mice lived. It didn't develop any bacteria. But a combination of the R, the rough strain, and the S strain, which is the smooth strain, when we put them together, the transformation principle what emerged from the heat-killed S bacteria, allowing them to transform into what? Smooth-coated bacteria and become what? Virulent. So these are the pictures of what our sister was demanding. So you have the rough strain, that is non-virulent, and the mouth lives. It was injected. Okay. Then we have the smooth strain, which is virulent, pathogenic. The rough strain is non-pathogenic. The smooth strain is pathogenic. So it is injected into the mouth. The mouse perishes, it dies. Now, heat killed smooth strain. It is injected into the mouse, and the mouse lives. Rough strain and heat killed smooth strain. Combination of these two, injected into the mouse, transformation occurs, the mouse dies. So this, please, those who have unmuted, they should mute for me, please. I'm getting feedbacks here. So this is Griffith's experiment discovering the transforming principle in pneumococcus bacteria, streptococcus pneumonia, pneumococcus bacteria. So Avery, McCarthy, and MacLeod identifying the transforming principle. In 1944, 3 Canadian and American researchers, Oswald Avery, McLean McCarthy, and Colin MacLeod, set out to identify Griffith's transforming principle. These 3 scientists decided to have a test case on what we just talked about, Griffith's transforming principle. So to do so, they began with large cultures of heat-killed S cells, and those through a long series of biochemical setups determined by careful experimentation, progressively purified the transforming principle by washing away, separating out, or enzymatically destroying the other cellular components. By this method, they were able to obtain small amounts of highly purified transforming principle, which they could then analyze through other tests to determine its identity. Several lines of evidence suggested to Avery and his colleagues that the transforming principle might be DNA. The purified substance gave a negative result in chemical tests known to detect what proteins, but a strongly positive result in a chemical test known to detect DNA. The purified substance gave a negative result in chemical tests, but a strongly positive result in a chemical test known to detect what DNA. So you see the contradiction. So the elemental composition of the purified transforming principle closely resembled DNA. The elemental composition of the purified transforming principle, it closely resembled DNA. That is why the S, which was subjected to heat and mixed with R, closely resembled DNA in its ratio of nitrogen and phosphorus. So protein and RNA degrading enzymes have little effect on the transforming principle. But enzymes able to degrade DNA eliminated the transforming activity. So these results all pointed to DNA as the likely transforming principle. DNA is a likely transforming principle. However, Avery was cautious in interpreting his result because those days it was a bit jittery. If you are not careful, a lot of people would just rubbish. Look at the time it took for Mendel's work to be accepted. It took him 34 years after publication before Schmack and von Vries and the others looked into it, discovered his findings. So he realized that it was still possible that some contaminating substance present in small amounts, not DNA, was the actual transforming principle. So because of this possibility, debate over DNA's role continued until 1950, when Alfred Hershey and Martha Chase used a different approach to conclusively identify DNA as the genetic material. So in here, you still have the S strain, which has been denatured by heat. Then you have lipids, which is in sugars, as in the test tube. Okay, then you add proteinase. You add proteinase. So there's no protein here. And the arrow leading to the beaker says you need to add R cells. R cells. In the first place, you subjected, you have subjected that to heat. The S cells have been subjected to heat. Then you add proteinase as well as the R cells. So you have S cells appearing. S cells appearing after they have been killed, because you added proteinase, which is an enzyme, and you added it to the R cells. You realize that the S cells starts appearing. This is where the streptococcus starts emerging. That was why the mouse or the mice died in that regard. So the third set, the second set, you add RNase. RNase. And RNase is a RNA. Okay, no RNA being added. No RNA being added. Then you add R cells to the beaker. And then still you see that S cells are appearing. But in the third test tube, when you add DNase, that means no DNA has been added. But when you add R cells to it, you realize that there are no smooth cells appearing. So in the tube, you realize that S cells appear. That was why the mice died. You remove lipids and sugars from a solution of heat-killed smooth cells. Proteins, RNA, and DNA remain. Then you treat solutions with enzymes to destroy proteins, RNA, and DNA. Add to culture containing living R cells. Observe for transformation by testing for the presence of virulent S cells. So Avery, Macleod, and McCarthy's experiment on the DNA as genetic of a cell, and this is how it goes. So the Hershey-Chase experiment. This other one, in their now legend experiment, Hershey and Chase studied stereophage or viruses that attack bacteria. So the phages they used were simple particles composed of protein and DNA, with the outer structures made of protein and the inner core consisting of DNA. Hershey and Chase knew that the phages attached to the surface of a host bacterial cell and injected some substance, which was either a protein or DNA, into the host. This substance gave instructions that caused the host bacterium to start making lots and lots of phages. In other words, it was the phages genetic material. Before the experiment, Hershey thought that the genetic material would prove to be protein. To establish whether the phage injected DNA or protein into host bacteria, Hershey and Chase prepared 2 different batches of phage. In each batch, the phage were produced in the presence of a specific radioactive element, which was incorporated into the macromolecules as DNA and protein that made up the phage. One sample was produced in the presence of 35 S strains, a radioactive isotope of sulfur. So sulfur is found in many proteins and is absent from DNA. So only phage proteins were radioactively labeled by this treatment. 35 sulfur isotope, a radioisotope of sulfur 35. So the other sample was produced in presence of 32 P, a radioactive isotope of phosphorus. And phosphorus is found in DNA and not in proteins. So only phage, that is deoxyribonucleic acid, and not phage proteins, was radioactively labeled by this treatment. So in short, you realize that there were two types of isotopes. The sulfur isotope, which is 35, and the phosphorus isotope, which is 32, were used in Hershey-Chase experiments. So each batch of phage was used to infect a different culture of bacteria. So you inject sulfur 32 and sulfur 35, sorry, and phosphorus 32 into a particular culture of the experiment of the strain. So after infection had taken place, each culture was boiled in a blender, removing all remaining phage and phage parts from the outside of the bacterial cells. So finally, the cultures were centrifuged or spun at high speeds to separate the bacteria from the phage debris. Centrifugation, it causes heavier material such as bacteria to move to the bottom of the tube and form a lump called pellet. Lighter material such as the medium used to grow the cultures along with phage parts remains near the top of the tube and forms a liquid layer called the supernatant. It forms a liquid layer referred to as supernatant. So all this informs the Hershey-Chase experiment. There were two main types of isotope: sulfur 35 and phosphorus 32, and they were infected with different cultures of bacteria. And after infection had taken place, it was put in a blender to take away various parts from the bacteria cells. So, on the review, Edric Griffith conducted a series of experiments using Streptococcus pneumoniae, and this is a type of bacteria, as well as mice. So he wasn't trying to identify the genetic material, but rather trying to develop a vaccine against pneumonia. So in his experiment, Griffith used two related strains. In his experiment, Griffith used two related strains of bacteria known as R and S, the R strain and the S strain. He concluded that the R strain bacteria must have taken up what he called transforming principle, because they have rough ends from the heat-killed S bacteria, which allowed them to transform into smooth-coated bacteria and become virulent. So Oswald Avery, McLean McCarthy, and Colin MacLeod set out to identify Griffith's transforming principle. They began with large cultures of heat-killed S cells and through a long series of biochemical steps, purified, transforming principle by washing away, separating out enzymatically, destroying the other cellular components. That result all pointed to DNA as the likely transforming principle. So Hershey and Chase then studied bacteriophage, or viruses that attack bacteria. So the phages they used were simple particles composed of protein and DNA, with the outer structures made of protein and the inner core consisting of DNA. So Hershey and Chase injected some substance, which was either DNA or protein, into the host. This substance gave instructions that caused the host bacterium to start making lots and lots of phages. In other words, it was the phages genetic material. Thank you. Unit 2, Session 3. Unit 2, Session 3. Structure and function of DNA. Structure and function of DNA. So at the end of this session, you should be able to explain in detail the structure of the deoxyribonucleic acid molecule and relate the structure of the DNA molecule to its function. You should be able to thoroughly explain in detail the structure of DNA molecule. You relate the structure to its function. A DNA molecule consists of two complementary chains of nucleotides. So a DNA molecule consists of two long polynucleotide chains composed of 4 types of nucleotide subunits. Each of these chains is known as DNA chain or a DNA strand. Hydrogen bonds between the base portions of the nucleotides hold the two chains together. Nucleotides are composed of a 5-carbon sugar to which are attached 1 or more phosphate groups and a nitrogen-containing base. In the case of the nucleotides in DNA, the sugar is deoxyribose attached to a single phosphate group. Hence, the name deoxyribonucleic acid. And the base may be either adenine, cytosine, guanine, or thymine. So, at the end of the day, you realize that the DNA nucleotide has a sugar component which is attached to a phosphate. And hence, the base, the nitrogenous bases, we have sugar, we have phosphate, and nitrogenous bases. And the bases are either adenine, cytosine, guanine, or thymine. And they are pairings, complementary pairings. We'll be looking at that very soon. So the nucleotides are covalently linked together in a chain through the sugars and the phosphates, which thus form a backbone. The sugar and the phosphates form the backbone of the DNA, while the complementary base pairings are alternate in that order. So they form a backbone of alternating sugar, phosphate, sugar, phosphate. So because only the base differs in each of the, each polynucleotide chain in DNA is analogous to the necklace as a backbone strand with 4 types of beads. The 4 bases are A, C, G, and T, adenine, cytosine, guanine, and thymine, often replaced by uracil in ribonucleic acids. So these symbols, same symbols, A, C, G, and T, are also commonly used to denote the 4 different nucleotides that the bases with their attached sugar and phosphate groups. So in here, you could see the building blocks of DNA. You have a phosphate sugar, then you have a base. The base is G, G representing guanine. So the nucleotide, you see the sugar phosphate sitting on the nucleotide. So it is attached to it because that is where the backbone lies. That is where the backbone lies. So the double-stranded DNA has a sugar phosphate, right? The sugar phosphate that has some brownish, light brownish in there with the golden spots that has been lined. And you have your complementary base pairings. You have G, T, A, T, C, sorry. Then you have B, G, B, C, and A. And G always pairs with C. A always pairs with T. T always pairs with A. And C always pairs with B. In that mode of fashion. And they are held together by hydrogen bonds. They are held together by hydrogen bonds. So you see the double-stranded DNA, which has one. But DNA double helix. You see a helix. It looks like the figure of 8. That's how it looks like. DNA and its building block. The sugar, phosphate, you have your base pairings, your nitrogenous base pairings. So a DNA molecule consists of 2 complementary chains of nucleotides, forming the giant double-stranded helix structure. So the way in which the nucleotide subunits are lined together gives a DNA strand a polarity. So if we think of each sugar as a block with a protruding knob, 5' phosphate on one side of the whole, and the 3' hydroxyl on the other side. See if figure 1, for instance, each completed chain formed by interlocking knots with holes will have all
Hello. Hello.
I can't hear anything anymore. What is going on?
Okay. So no problem. My screen is off.
Yeah, dock is off.
Okay.
Oh, okay. Okay.
The screen is no more showing.
Flowing, yes. And the talking
Only,
to you is not.
only the picture.
Yeah.
Maybe he might not be aware to.
Yeah, maybe.
He might be talking like that, but he might not be aware we are not.
Hmm.
Is he more on the page?
Hello? Do you hear me now?
Hello, Doc. Yes, we can hear you.
Don't mind me, and don't mind the network. The communication minister, I'll talk to him so that we would receive in such a way. That's just by the way. That's just by the way. So sorry for the disconnection. So the three-dimensional structure of DNA, that's the double helix, arises from the chemical and structural features of its polynucleotide chains. Because these two chains are held together by hydrogen bonding between the bases on the different strands, and all the bases are on the inside of the double helix, and the sugar phosphate forms the backbone, just like what we saw in the earlier figure. So in each case, a bulkier two-ring base, a purine, is paired with a single ring base, which is pyrimidine. So A, which is adenine, always pairs with thymine. So A is a purine. It always pairs with the pyrimidine thymine. And G, which is also a purine, always pairs with the pyrimidine cytosine. So this complementary base pairing enables the base pairs to act in an energetically most favorable arrangement in the interior of the double helix. So their backbone is normally the sugar and phosphate, whilst the inside of it, the nitrogenous bases, which are the purines and the pyrimidines, always form the bonds held together by hydrogen bonds. So in this arrangement, each base pair is of similar width, thus holding the sugar phosphate backbones an equal distance apart along the DNA molecule. So to maximize the efficiency of base pair packing, the two sugar phosphate backbones wind around each other to form a double helix with one complete turn every 10 base pairs. So in here we have the double helix structure. We have a minor groove and a major groove. The difference is that one is not so much packed. The major groove, you realize that it is thoroughly packed. Now let's look at the 5' and 3' terminals, the endings. Okay, the hydroxyl and the phosphate. That's forming the backbone. Okay, so you realize that the bases, the purines and the pyrimidines, you have adenine and guanine, adenine and guanine, G and C interacting there. Then you see C and G in the next, in that order. But you realize that the 3' end, that is the hydroxyl end, the phosphate, you realize that it has lots of what? Oxygen that attached to the phosphate prime, the phosphate end. So those ends are quite different. One signifying a prime, a 3' end is different from one signifying a 5' end. You realize that because it is 3', you have what? 3 base pairs. And with the 5', you realize that the phosphate is in between what? 5. There are 5 what elements? Phosphorus and 4 oxygen molecules that are what? Attached to it. So that informs the 3' and the 5'. So you see at one end, you realize that 5', you have phosphorus attached to 2 bonds of oxygen having 2 bonds being attached to phosphorus, then the phosphorus being attached to single bonds of what? Oxygen, and there is another oxygen sitting on the backbone, So So this informs the DNA double helix structure. Double helix structure. Purines pairing with pyrimidines, with their backbone being the sugar phosphate molecules, forming the backbone of the double helix structure, DNA double helix. So, better still, a more comprehensive structure of what we looked at earlier. On top, we have the 3', the hydroxyl, okay, the 3'. And you realize that at its end, you have 5'. So 5' starts and 3' goes in that direction. So you have thymine pairing with what? Adenine. And cytosine pairing with what? Guanine. D pairing with what? C. And T pairing with what? E. Adenine. The molecule adenine, the nitrogenous base adenine. In a double helix structure. So a DNA molecule consists of, yes.
Doc, please, I wanted to know the 5 and 3, what they signify.
The 5', you'd realize that the purines pick up the phosphate molecules, and they stand for the 5. They have 5 base pairings, okay? And the pyrimidines, that has to do with the hydroxyl group, and that deals with 3 base pairings. Is that all right?
Okay, Doc, thank you.
You're welcome. So I believe I can continue.
Yes, Doc.
Very well. So shapes and chemical structure of the bases allow the hydrogen bonds to form efficiently only between adenine and T, just as you were asking, between guanine and cytosine. So where atoms that are able to form hydrogen bonds can be brought close together without distorting the double helix. As indicated, 2 hydrogen bonds form between adenine and thymine. Adenine and thymine. So that informs what? The pyrimidines. While 3 form between what? Guanine and cytosine. 2 hydrogen bonds form between adenine and thymine, while 3 bonds form between what? Guanine and cytosine. So the base can pair in this way only if the 2 polynucleotide chains that contain them are anti-parallel to each other. So if they are not parallel, they wouldn't what? Form any bond. If there are no complementary base pairings, they wouldn't pair. They wouldn't pair. Is that right? Nature has its way of what? Pairing up. Then excuse me. Some human beings are claiming they want to be married to LGBTQQ++, right? They are of a different view altogether, even with regards to enzymatic reactions. DNA have their base pairings. Without it, it cannot pair. Without it, it cannot pair. So members of each base pair can fit together within the double helix. Only if the 2 strands of the helix are anti-parallel. That is, only if the polarity of one strand is oriented opposite to that of the other strand. So a consequence of these base pairing requirements is that each strand of a DNA molecule, the double helix structure contains a sequence of what? Nucleotides. That is exactly complementary to the nucleotide sequence of its partner strand. So one nucleotide, when a double helix unwinds, you would have the backbone, the phosphate sugar backbone, and complementary base, nitrogenous base pairings. Okay, and they will have their complementary on the other hand. So when these come together, they unwind, form a double helix structure. So the base pairing between the complementary strands is required for two important functions of DNA. The pairing between the complementary strands is required for two important functions of DNA. And what are the two main important functions of DNA? DNA replication. Replication simply means doubling. DNA, it can double itself, just like the cell amoeba proteus. When it meets favorable conditions, they are able to what? To multiply in no time. Just like some viruses, when they are outside, they act as particles, and immediately they get into a living organism, they start replicating. So DNA replication involves an unwinding of the double helix, followed by synthesis of a complementary strand from each of the unpaired template strands. And DNA is serving as a template for RNA synthesis. It is out of a DNA that RNA emerges. It is out of a DNA that the RNA emerges. So it serves as a template for RNA synthesis by utilizing the information in one strand to code for a complementary RNA strand. Now let's look at the structure of DNA providing mechanism for heredity. So genes carry biological information that must be copied accurately for transmission to the next generation. Each time a cell divides to form two daughter cells, two central biological questions arise from these requirements. So how can the information for specifying an organism be carried in chemical form, and how is it accurately copied? The discovery of the structure of DNA double helix was a landmark in the 20th century biology because it immediately suggested answers to both questions, thereby resolving at the molecular level the problem of heredity. So we discussed briefly the answers to these questions in this section, and we shall examine them in more detail in subsequent chapters. So DNA encodes information through the order or sequence of the nucleotides along each strand. Each base, that is A, C, T, or G, adenine, cytosine, thymine, or guanine, can be considered as a letter in a four-letter alphabet that spells out biological messages in the chemical structure of the DNA. Again, organisms differ from one another because their respective DNA molecules have different nucleotide sequences, and consequently it carries out different biological messages. But how is the nucleotide alphabet used to make messages, and what do they spell out? That is the multi-million-dollar question. So, as discussed, it was known well before the structure of DNA was determined that genes contain the instructions for producing proteins. The DNA messages must therefore somehow encode proteins. This relationship immediately makes the problem easier to understand because of the chemical character of proteins. So We shall discuss that in later units. The properties of a protein, which are responsible for its biological function, are determined by its three-dimensional structure. And its structure is determined in turn by the linear sequence of the amino acids of which it is composed. The linear sequence of nucleotides in a gene must therefore somehow spell out the linear sequence of amino acids in a protein. The exact correspondence between the four-letter nucleotide alphabet of DNA and the 20-letter amino acid alphabet of proteins, the genetic code, is not obvious from the DNA structure. And it took over a decade after the discovery of the double helix before it was worked out. So, a figure, the relationship between genetic information carried in DNA proteins. So you have three types of genes: gene A, gene B, gene C. And the gene expression, you have double helix structure. The gene expression is that you have protein A, protein B, and protein C. Relationship between genetic information that is carried in DNA and protein. So depending on which gene and the information that is carried, the gene expression that it carries. So in review, nucleotides are composed of a 5-carbon sugar to which are attached one or more phosphate groups and a nitrogen-containing base. So in the case of the nucleotides in DNA, the sugar is deoxyribonucleic. Sorry, let me do that again. In the case of nucleotides in DNA, the sugar is deoxyribose attached to a single phosphate group, hence the name deoxyribonucleic acid. And the base may be either adenine, cytosine, guanine, or thymine. The nucleotides are covalently linked together in a chain through the sugars and phosphates, which thus form a backbone of alternating sugar-phosphate molecules. So genetic information, on the other hand, is carried in linear sequence of nucleotides in DNA. Each molecule of DNA is a double helix formed from two complementary strands of nucleotides held together by hydrogen bonds between guanine and cytosine and adenine and thymine base pairs. Duplication of the genetic information occurs by the use of one DNA strand as a template for formation of a complementary strand. The genetic information stored in an organism's DNA contains the instructions for all proteins the organism will ever synthesize. In eukaryotes, DNA is contained in the cell nucleus. In eukaryotes, it is contained in the cell nucleus. That brings us to the end of Unit 2, Session 3. Unit 2, Session 3. It looks as if today our sessions are not as lengthy as what we started with last week. Now let's move on to Unit 2, Session 4, the chemistry of DNA. The chemistry of DNA. So at the end of this session, you should be able to understand the behavior of DNA molecule when it interacts with other elements. You should be able to understand the behavior of DNA molecule when it interacts with other elements. And again, explain the forces that affect the stability of the DNA molecule. Hello? Do you hear me loud and clear?
Yes, sir. Yes, sir.
Yes, sir.
Very well. All right. So the session objectives are that you need to understand the behavior of DNA molecule when it interacts with other elements, and again explain the forces that affect the stability of the DNA molecule, the stability of DNA. So now let's look at the chemistry of DNA. What actually goes into the formation of DNA? Forces affecting the stability of the DNA molecule. So forces affecting the stability of the DNA molecule. There are 4 types of forces contributing to maintaining the stability of the DNA double helix. And these include the hydrophobic interactions, stacking interactions,
Hello, Doc.
hydrogen bonding. Yes.
Please, it seems some echo is at your background.
Very well. Please, just give me a few, then I'll quickly relocate and get back to you. Let's pause for about 5 minutes. Okay.
Thank you, Doc.
Very well. Thank you. Hello. Hello. Please,
Hello,
do you
Doc.
hear me now?
Yes,
Please
sir.
talk.
Very well. I
Yes,
believe
God.
it's now better. I believe it's now better.
Yes.
Yes,
Yes,
please.
All right.
everything is clear now.
Very well. Please, let's continue. Sorry for the hold up. So let me begin again by saying forces affecting the stability of the DNA molecule. So there are certain forces affecting the stability of the DNA molecule, and there are 4 types contributing to maintaining the stability of the DNA double helix structure. And it includes the hydrophobic interactions, that is one, the stacking interactions, the hydrogen bonding, as well as electrostatic interactions, Now, let us move on. So, the hydrophobic interactions do stabilize the DNA molecule. They remain hydrophobic inside and hydrophilic outside. Inside, it fears water, but outside, it loves water. Hydrophobic. Phobia, the fear of water. Hydrophilic, outside. Right. So stacking interactions also stabilize the DNA molecule. Stacking, putting things together, stacking interactions. They are relatively weak, but additive van der Waals forces. They are actively weak. They are relatively weak by additive van der Waals forces. That is what puts them together. Then hydrogen bonding also stabilizes the DNA molecule. They are also relatively weak, but additive and facilitate stacking. So electrostatic forces of interactions destabilize the DNA molecule. So you see, you are hearing hydrogen bonding, van der Waals forces, and other interactions that involves the chemistry of what? DNA, electrostatic forces, van der Waals forces, hydrogen bonding. So they destabilize the DNA molecule. That's the electrostatic interactions. And they are primarily they are contributed primarily by the negative phosphates. The negative phosphates, the 5 prime. They affect intra-strand, the DNA strand, those within, strand by strand, interstrand and interstrand interactions. Okay, then repulsion can be neutralized with positive charges. That's positively charged sodium ions or proteins. So the base pairs, earlier we made those submissions, the purines and the pyrimidines, adenine, guanine, cytosine, and thymine. Okay, the base pairs, they are forming the base pairs. So the base pairs in the interior of the DNA molecule create a hydrophobic environment. So the nitrogenous bases they do not like water. So they create a hydrophobic environment with the negatively charged phosphates along the backbone being exposed to the solvent. So the backbone, as for that, is so they don't care. It's hydrophobic. And so they love water. But within it, it doesn't. So thus, in an aqueous environment, the double-stranded structure is stabilized by the hydrophobic interior. It's stabilized by the hydrophobic interior. Now, let's look at reagents that solubilize the DNA bases. Reagents, a certain chemical that makes DNA soluble. And the first example is methanol. Methanol, it stabilizes the double helix. Stacking interactions and hydrogen bonding interactions relatively weak, but additive. So the reagents that disrupt hydrogen bonding, example, formaldehyde, urea, and solutions with very low pH, which is less than what, 2.3, or very high pH, greater than 10, stabilize the double helix. So electrostatic repulsion, they repel by negatively charged phosphate along the DNA backbone, destabilize the double helix. Destabilize the double helix. Neutralizing these negative charges by the addition of sodium chloride, which contributes positively charged sodium ions to the DNA solution, will prevent strand separation. In the cell, the phosphates also interact with positively charged ions, magnesium, potassium, or sodium, and with positively charged basic proteins. So in here, base stacking and charge repulsion in the DNA double helix. So you have the charge repulsion. The backbone repels it, and the stacking interactions occurring in the interior of the double helix. So deoxyribonucleases, these are enzymes. Anytime you come across any word ending in ase, A-S-E, hydrogenase, you should know that it is an enzyme. Deoxyribonucleases or DNases are enzymes that cleave phosphodiester bonds. They cleave phosphodiester bonds. Deoxyribonucleases, they cleave, they get weaved, they get hooked onto phosphodiester bonds. Bonds that are rich in what? Phosphorus. The 5' that we talked about earlier. Okay, so deoxyribonucleases are enzymes that cleave, they get attached to phosphodiester bond. And some are used for constructive purposes, such as proofreading during DNA replication, whereas others are used to degrade DNA. So there are two basic classes of deoxyribonucleases: exonucleases and endonucleases. Exo simply means out, and endo simply means in. So exonucleases remove only the terminal nucleotide, whereas endonucleases cleave endo, inside. They cleave anywhere within the DNA double helix. Now, deoxyribonucleases can be denatured. They can be destroyed when exposed to harsh conditions such as extreme temperatures of heat and hydrogen ion concentration, pH. So therefore, stabilizing the DNA double helix can become can be overcome by heating the DNA in solution or by treating it with very high or very low pH. Low pH will also damage the DNA, whereas high pH will simply separate the polynucleotide chains. So once denaturation has begun, there is a cooperative unwinding of the double helix that ultimately results in complete strand separation. So when the strands of DNA separate, DNA is said to be denatured. When the strands of DNA separate, the DNA is said to be denatured. That means that when separation comes in, the strands will be denatured, they will be destroyed. So when the high temperature is used to denature DNA, the DNA is said to be what? Melted. Because some of the forces stabilizing the DNA double helix are contributed by base pairing interactions. And because adenine pairs with thymine, A-T base pairs have only 2 hydrogen bonds in contrast to GC base pairs, which have 3 hydrogen bonds. Regions of DNA duplex that are AT rich will denature first because they have bonds, 2 bonds. 2 bonds. It is easier to break through 2 than 3. So they would be denatured first. They'll be destroyed first before getting to the guanine and cytosine base pairings.
Hello, Doc.
Yes.
Please, with the denaturing of the DNA,
Yes.
will it happen or can it happen in living organisms when they are alive?
It can. It can. It can. You know, let me cite examples, typical examples. That is why you are advised not to be sleeping with your mobile phones, electronic gadgets on whilst you sleep. The rising cases of cancers is as a result of some of these things. They denature the cells, okay, inherent in us, and they become cancerous. And at the end of the day, you are dying slowly.
Oh, okay.
So that is the way to go. It doesn't necessarily mean you being exposed to high amounts of what heat, and even too much X-rays. Okay, too much exposure to X-rays can also lead to denaturing of some of these DNAs.
Oh, okay. So, sir, can it also happen that the purine alone can pair?
No. It always has to pair with a pyrimidine.
Oh, okay.
Yes.
Right.
It always
Thank
has to
you,
pair
Doc.
with a yes. So you have the double-stranded DNA, then the strand separation occurs, okay? And formation of single-stranded pairs. You see the single-stranded, right? So single-stranded, the double-stranded DNA, and we move down to extremes in pH or high temperature. That is what is going to bring about what? The denaturing. So it will segregate. It will start what? Breaking. If you expose it to extreme low pH or high pH, highly acidic or highly basic or highly alkaline solutions, that is when it breaks the what? The hydrogen bonds holding the base pairings. So cooperative unwinding of the DNA starts when they are exposed to extreme temperatures or extreme pH. So the unwinding occurs until you have what? The single strands. And the single strands will then reform into another complementary base pairing, forming a double helix structure. It will definitely look out for the same complementary base pairings to align itself to it, reform the base pairings. That was why in the earlier submission, Avery and McLeod, okay, you realize that when the S strain and the R strain, the S was injected, the mice died. But when R was injected into the mice, it didn't die. But a combination of the two that had been exposed to high levels of what heat, and it has denatured the S strain, realized that the S started what? Transform, and the mice developed pneumonia and died. So these bears the stages of the process of denaturing of the DNA double helix structure. So over here, it still goes on to talk about it. A double-stranded guanine cytosine-rich DNA has not yet melted. Then, AT-rich region of DNA has melted into a single-stranded bubble. Single-stranded bubble. So AT-rich regions melt first. Why? Why would it melt first? Because it contains what? Only two bonds. It is held by only two what? Hydrogen bonds. So it will melt first, or it will break or unwind first before the what? Double-stranded DC-rich DNA will melt, because it has what? 3 bonds, hydrogen bonds. So that is how the unwinding takes place. That is how the unwinding takes place. Hyperchromicity. 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 This is when a solution of double-stranded DNA is placed in a spectrophotometer cuvette, and the absorbance of the DNA is determined across the electromagnetic spectrum. So it characteristically shows an absorbance maximum at 260 nanometers in the UV region of the spectrum, 260 nanometers in the ultraviolet region of the spectrum. So if the same DNA solution is melted, the absorbance at 260 nanometers increases approximately 40%. So this property is termed hyperchromicity. If the same DNA solution is melted, the absorbance at 260 nanometers increases approximately 40%. So this property is termed hyperchromicity. So the hyperchromic shift is due to the fact that unstacked bases absorb more light than stacked bases. The hyperchromic shift is due to the fact that unstacked bases absorb more light than stacked bases. Absorbance maximum for single-stranded DNA. Absorbance maximum for single-stranded DNA. Absorbance maximum for double-stranded DNA. So the absorbance of 220, 260, and 300. You realize that the absorbance is high at 260 and declines at 300. So the absorbance at 260 nanometers of a DNA solution increases when the double helix is melted into single strands. So hyperchromicity can be used to follow the denaturing of DNA as a function of increasing temperature. So as the temperature of a DNA solution gradually rises above 50 degrees Celsius, the adenine thymine regions will melt first, giving rise to an increase in the ultraviolet absorbance. So as the temperature increases, furthermore of the DNA will become single-stranded. further increasing the absorbance until the DNA is fully denatured above 90 degrees Celsius. The temperature at the midpoint of the melting curve is termed melting temperature and is abbreviated TM, melting temperature. So the TM for a DNA depends on its average GC, guanine and cytosine content. The temperature, melting temperature for a DNA depends on its average guanine and cytosine content. So the higher the guanine and cytosine content, the higher the melting temperature. So note that guanine plus cytosine content or GC content and GC content are equivalent terms. That is, they are used interchangeably. So wherever you see G plus C or G is to C or GC, it is still referring to the guanine content, the guanine cytosine content, guanine cytosine content. And don't forget, they are used interchangeably. So they could bring even some of these examples in your objective test items or multiple choice items for you to select from.
Doc,
Yes.
in that case, it seems the way it is a little challenging. So can you guide us with guiding questions so we know the area to study, how to go about it?
No. I'm just raising assertion that you see, you are being prompted that G plus C content simply means GC content or GC. You realize that G plus content, comma G dash C content and GC. It is still referring to the same content, guanine cytosine content.
Yes, Doc, I understand
depending on the author, right? Depending on the author. That is why I made emphasis to the fact that you could even meet such a case in your multiple choice exams. For instance, we are giving the range of base pairings. Select which of the base pairings is right. You could have AG, AC, GC, and CC. Which of them is right? And per your knowledge with regards to base pairings, you should always remember that A always pairs with T and G always pairs with C. There is no way A is going to pair with G and C going to pair with C, or A pairing with A. Is that right? So that includes what is bothering your mind. Can I go on? Now let's look at DNA renature or association. Denaturing of a DNA double helix produces single-stranded DNA. So the reverse reaction, the formation of double-stranded DNA, can be carried out if the complementary DNA strands are incubated under conditions that will promote renaturation or reassociation. So the first step is slower. The first step in DNA renaturation is slow, in which the complementary strands attempt to find each other. The strands randomly interact until complementary base pairing can occur. This may occur over or only a short region of each strand, but may be enough to nucleate the reaction. Because the two reacting strands are at the same concentration in solution, the reaction is second order, the rate of the reaction being defined by the second order rate constant, that's K2. So once nucleation has occurred, the complementary strands zipper up, the zipper up. So it's just like you having a trousers. When you want to zip up, it closes. When you zip down, it opens. When you zip down, that it means what? Denaturing is taking place. Reassociation. So the complementary base pairs, when they are coming together, they are forming the double helix stranded structure. Is when you are zipping your trousers. When you zip it up, it means that you have closed it. They form the double helix structure. But when you zip down, then it means what? You are forming the single strands. So illustration of the process of protein reassociation. Protein reassociation. So denatured single-stranded DNA under K2. So slower rate limiting second order process of what? Finding complementary sequences to nucleate base pairing. So using the K2, you realize that there is a faster zippering reaction to form along molecules of double-strand DNA. So the single strands, you realize that it starts what? Pairing. Then it leads to what? The double-stranded DNA. A typical example is the zipper in our trousers, in our jackets that I made emphasis to. That is how reassociation or renaturing occurs in the DNA molecule or strands. Now, secondly, the complexity of a DNA is a function of how many base pairs it has. In other words, a low complexity DNA may consist of a few hundred or a few thousand base pairs. Low 100 to 1,000 base pairs. In contrast to high complexity DNA, that may contain millions of base pairs. So low complexity sequences are able to find each other much faster. Find each other much faster. Like you want to mix up squash in a cup and squash in, let's say, a bucket. You realize that the one within the cup, you'll be able to mix it faster, but the one within the bucket will take time. So low complexity sequences are able to find each other much faster during a renaturation reaction than our high complexity sequences, since the low complexity sequences have to make fewer coalitions to find a base pairing partner. So if it is short, it is easier to form a bond. The bond is easier to form easily rather than when it's very complicated. So thus, the rate of renaturation reaction is a function of the complexity of the DNA. This of a DNA can be determined by measuring its second order reassociation rate constant. The human genome consists of 3 populations of DNA. The first and intermediate fractions make up our 10% and 15% of the genome, respectively, and the slow fraction that makes up about 75% of the genome. Most of the genes in the human genome are in the single copy fraction. shown in the next slide. Repeated sequences can be of two types. Those that are interspersed throughout the genome, or those that are tandemly repeated satellite DNAs. So among the interspersed repeated sequences are so-called allele sequences that are about 300 base pairs and are repeated about 300,000 times in the genome. So you can imagine what goes on with regards to single copy fractioning, most of the genes in the human genome. So they can be found adjacent to or within genes, and presence can sometimes lead to the occasional disruption of genes. So the interspersed, those that have been passed into wide areas, the interspersed repetitive sequences also include variable number of tandem repeats, which are made up of short repeated sequences of only a few base pairs, but of variable lengths. They are also interspersed throughout the genome and are quite useful as landmarks for mapping genes because they are highly polymorphic. They differ in length or number of repeats from person to person. Now, in review, what types of forces contribute to maintaining the stability of the DNA double helix? They include hydrophobic interactions, stacking interactions, hydrogen bonding, and electrostatic interactions. And these affect intrastrand and intrastrand and interstrand interactions. So repulsion can be neutralized with positive charges. Example, positively charged sodium ions or proteins. The base pairs in the interior of the DNA molecule creates a hydrophobic environment with the negatively charged phosphates along the backbone being exposed to the solvent. Thus, in an aqueous environment, double-stranded structure is stabilized by the hydrophobic interior. So agents that solubilize the DNA bases, example, methanol, destabilize the double helix. Stacking interactions and hydrogen bonding interactions are relatively weak but additive. Reagents that disrupt hydrogen bonding, such as formaldehyde, urea, and solutions with very low pH, that is as low as 2.3, and very acidic, or very high pH, that's about 10. The alkalinity is very high. It destabilizes the double helix. Electrostatic repulsion by negatively charged phosphates along the DNA backbone destabilize the double helix. Deoxyribonucleases or DNases are enzymes that cleave phosphodiester bonds. They cleave onto the phosphodiester bonds, and some are used for constructive purposes, such as proofreading during the DNA replication, whereas others are used to degrade DNA. Again, DNA can be denatured when exposed to harsh conditions such as extremes of temperature and hydrogen ion concentration, that's pH. DNA can be reassociated if the complementary DNA strands are incubated under conditions that will promote renaturation. So that has to do with unit 2 session 4. So we are moving to unit 2, session 5. Unit 2, session 5. And that has to do with genes and genomes. Genes and genomes. Genes and genomes. So at the end of this session, our students should be able to understand the differences between genomes of prokaryotes and eukaryotes. Pro, before nucleus. Pro is before. Carrion is nucleus. Carrion is nucleus. And U is true. So they have, eukaryotes have a true nucleus. Explain genome composition and the structure of a gene. Explain genome composition and the structure of a gene. Structure and function of a gene. Genes are units of genetic information present on the DNA, the chromosomes, and chromatin of an organism. So the structure of a gene consists of many elements, of which the actual protein coding sequence is often only a small part. These include DNA regions that are not transcribed, as well as translated regions of the RNA. Structure and function of a gene. Structure and function of a gene. Planting the open reading frame. Genes contain a regulatory sequence that is required for their expression. So first, genes require a promoter sequence. And the promoter is recognized and bound by transcription factors and RNA polymerase to initiate transcription. So the recognition typically occurs as a consensus. We need to agree to disagree. A consensus sequence like the Tata box, a gene can have more than one promoter, resulting in messenger RNAs that differ in how far they extend in the 5' end. So you have 3 types of RNAs: the messenger RNA, ribosomal RNA, as well as transfer RNA. So highly transcribed genes have strong promoter sequences that form strong associations with transcription factors, thereby initiating transcription at a high rate. Other genes have weak promoters that form weak associations with transcription factors and initiate transcription less frequently. So eukaryotic promoter regions are much more complex and difficult to identify than prokaryotic promoters. So additionally, genes can have regulatory regions, many kilobases upstream or downstream of the open reading frame that alter expression. So these act by binding to transcription factors, which then cause the DNA to loop so that the regulatory sequence and bound transcription factor become close to the RNA polymerase binding site. So example, enhancers or promoters increase transcription by binding an activator protein, which then helps to recruit the RNA polymerase to the promoter. Conversely, silencers bind repressor proteins and make the DNA less available for RNA polymerase. So the transcribed pre-messenger RNA, messenger is coming from the unwound double-stranded helix structure. So the transcribed pre-messenger RNA contains untranslated regions at both ends, which contain a ribosome binding site, terminator, and start and stop codons. So in addition, most eukaryotic open reading frames contain untranslated introns, which are removed from the exons are translated. Because initially they were together, one is removed from the other. So one becomes an intron. In means it is entering. Then extron means it is moving out. Okay. So in addition, the most eukaryotic open reading frames contain untranslated introns, which are removed before the exons are translated. So the sequences at the ends of the introns dictate the splice sites to generate the final mature mRNA, which encodes the protein or RNA product. So this mRNA, in a nutshell, moves from the nucleus into the cytoplasm to undergo protein synthesis.
Hello, sir.
In the nucleus, yes.
Hello, sir.
Yes, I'm here.
Sir, good morning once again. I want to ask, when can we get a 10 or 15 minutes break?
You want to get 10 or 15 minutes break?
Yes.
Okay, so after in the next 5 minutes, okay.
All right, sir.
Very well. Can I
Thank you.
continue?
Yes, please.
All right. Okay, so many prokaryotic genes are organized into operons. Operon simply means they are operators with multiple protein coding sequences that are transcribed as a unit. So the genes in an operon are transcribed as a continuous messenger RNA, referred to as a polycistronic messenger RNA. So the term cistron in this context is equivalent to gene. The transcription of an operon's mRNA is often controlled by a repressor that can occur in an active or inactive state depending on the presence of certain specific metabolites. When active, the repressor binds to a DNA sequence at the beginning of the operon called the operator region and represses transcription of the operon. When the repressor is inactive, transcription of the operon can occur. The products of operon genes typically have related functions and are involved in the same regulatory network. Figure 5.1 illustrates the generalized structure of a gene. So this is the generalized structure of a gene, where we have the promoter region, you have exons. Exons are filled and unfilled boxes regions. Okay. Then the introns, they are between the exons. Introns, they are between the exons. Transcribed region. So mRNA, you see the 5 prime, the 3 prime. Generalized structure of eukaryotic gene. Generalized structure of a eukaryotic gene. So let's pause here so that when we come, we continue from gene structure and organization. Is that all right?
So when are we coming back?
Very well, sir.
In
Which
the next
time are
10
we
minutes.
returning back?
In the next 10 minutes. That should be around 9:20, right? That should be around 9:20.
Okay.
Is that
Okay,
right?
Sir, please
Agree.
make it 9:30.
Come again.
Sir, please give us about 30 minutes.
About 30 minutes is saying, hey,
Oh, sir.
Quarter
we
by
we
9:30 we should
no, no,
return back.
no. We will finish
By
very soon.
9:30.
We will finish the lecture very soon. Today's lecture is not lengthy, unlike last week's. And so I would have even wished we finish everything before you go on your break. But since you are demanding for the break, let's use the 10 minutes. Is that right?
Okay, thank
So
you.
maximum 15 minutes. Maximum 15 minutes.
Okay.
We'll return at
Thank you.
9:25, please. Most welcome.
Thank you.
Okay, sir. Okay, sir. Thank you.
Welcome.
Hello, sir.
Pedagua.
Please, my network is misbehaving. I'm not getting.
Hold the opening camera. You dey leave, eh?
Welcome back. And I believe you can see the screen.
Yes, yes, please. Everything is clear.
All right, genome structure and organization. So we are continuing from where we left off. So genome is all the DNA contained in an organism or a cell. Genome, genes, that makes up the entirety of an organism. Genome is all the DNA contained in an organism or a cell, which includes the chromosomes plus the DNA in mitochondria and DNA in the chloroplast of plant cells. So the number of genome sequenced in their entirety is now in the thousands and includes organisms ranging from bacteria to mammals, prokaryotes through to eukaryotes. So the first complete genome to be sequenced was that of the bacterium Haemophilus influenzae in 1995. Haemophilus influenzae. So the term genome can be applied specifically to mean that what is stored on a complete set of nuclear DNA, that is the nuclear genome, but can also be applied to what is within organelles, that contain their own DNA, as with the mitochondrial genome or the chloroplast genome. Additionally, the genome can comprise non-chromosomal genetic elements such as viruses, plasmids, and transposable elements. Typically, when it is said that the genome of a sexually reproducing species has been sequenced, it refers to a determination of the sequences of one set of autosomes and one of each type of sex chromosome, which together represent both of the possible sexes. Even in species that exist in only one sex, what is described as a genome sequence may be a composite read from the chromosomes of various individuals. Colloquially, the phrase genetic makeup is sometimes used to signify the genome of a particular individual or organism. The study of the global properties of genomes of related organisms is usually referred to as genomics, which distinguishes it from genetics, generally studies the properties of single genes or groups of genes. Genome structure and organization. So an analogy to human genome stored on DNA is that of instructions.
Dog, please. So, in a nutshell, I want to find out what's the difference between a gene and a genome.
A genome is actually a composition of all the genetic makeup of an organism, whilst a gene is a trait bearing in an organism. So all the DNA contained in an organism or a cell, including the chromosomes, plus that of the mitochondria, is referred to as what? A genome. And it is in both plants and animals. Is that right?
Thank you, sir.
Can I continue?
Yes, sir.
All right. So the analogy to human genome stored on DNA is that of instructions stored in a book. So the book genome would contain 23 chapters. 23 chapters signifying what? Chromosomes. All humans have what? 23 chromosomes. So each chapter contains 48 to 250 million letters. And the letters, as we discussed earlier, purines and pyrimidines, adenine, cytosine, guanine, thymine, A, C, G, and T without spaces. So hence the book contains 3.2 billion letters in total. The analogy with regards to genome structure and organization is equated to that of like a book with 23 chapters. And the 23 chapters is signifying the 23 chromosomes. Is that right? And the book fits into a cell nucleus. You wouldn't find genomes or genes in the cytoplasm of a cell. You would always find it in the nucleus. The nucleus, that is where you find what? The genes, the chromosomes. So at least one copy of the book is contained in most cells of our body, and the only exception in humans is found in mature red blood cells, which become denucleated during development and therefore lacks a genome. So genome composition. Genome composition is used to describe the makeup of contents of a haploid genome, which should include genome size, proportions of non-repetitive DNA and repetitive DNA in details. By comparing the genome compositions between genomes, scientists can better understand the evolutionary history of a given genome. Science can understand the evolutionary history of a genome. So when talking about genome composition, one should distinguish between prokaryotes and eukaryotes. Prokaryotes, bacterium, they have what? A nuclear material. But eukaryotes have a nucleus. They have two nucleus. As there are significant differences with content structure, there's a difference between bacterium cell and that of a plant cell or animal cell. So in prokaryotes, most of the genome, 85 to 90%, is non-repetitive DNA, which means coding DNA mainly forms it, while non-coding regions only take what? A small part. So bacterium cells do not have what? Nucleus, but they have what? Nuclear material. That means the non-repetitive DNA forms about 85 to 90%. 85 to 90%. So on the contrary, eukaryotes have the feature of exon-intron organization of protein coding genes. The variation of repetitive DNA content in eukaryotes is also extremely high. In mammals and plants, the major part of the genome is composed of repetitive DNA. Now let's look at the genome size. It is the total number of DNA base pairings, meaning cytosine, guanine, thymine, in one copy of a haploid genome. Haploid, 23 pairs of chromosome. So in humans, the nuclear genome comprises approximately 3.2 billion nucleotides of DNA, divided under 24 linear molecules. The shortest is 550 million nucleotides in length. The shortest, 50 million nucleotides in length, and longest, 260 million nucleotides, each contained in a different chromosome. So the genome size is positively correlated with the morphological complexity among prokaryotes and lower eukaryotes. However, after mollusk, animals with shells, like Achatina, the garden or the giant African snail, Achatina, whales, and other mollusks, and all the other higher eukaryotes above, this correlation is no longer effective. So this phenomenon also indicates the mighty influence coming from repetitive DNA act on the genomes. So there is experimental work being done on minimal genomes for single cell organisms, as well as minimal genomes for multicellular organisms. The work is both in vivo and in silico. It's still ongoing because it entails a lot to look. Now let's look at the different genomes of prokaryotes and eukaryotes. The genes of eukaryotes often have introns, intra-specific gene order, and number generally relatively stable. Many non-coding, that's RNA, ribonucleic acid genes. So there is not general relationship between organism complexity and gene number. Then in prokaryotes, there are no introns. Gene order and number may vary between strains of a species. Then the gene regulation promoters, often with distal long range enhancers or silencers, marked transcriptional domains, and generally monocestronic. With prokaryotes, they possess the promoters, they have enhancers as well as silencers, which are rare. Then genes often regulated as polycestronic operons, repetitive sequences, generally highly repetitive with genome, wide families from transposable elements, generally few repeated sequences, relatively few. That has to do with the repetitive sequences. And when it comes to size, it's as large as 10 megabytes or 100,000 megabits. Right. There is not generally a relationship between organism complexity and its genome size. Many plants have larger genomes than humans. Then when it comes to size with regards to prokaryotes, they are generally small, less than 10 megabits, mostly around 5 megabits. Their complexity, as measured by the number of genes and metabolism, generally proportional to genome size. Then content, most DNA is non-coding. DNA is coding gene dense. Then with telomeres or centromeres, with eukaryotes, it is present. In prokaryotes, it is circular DNA because they don't possess a true nucleus. It doesn't need telomeres, don't have mitosis, hence no centromeres. The number of chromosomes in eukaryotes are more than one, often including those discriminating sexual identity. Then with prokaryotes, often one, sometimes more, but plasmids, not true chromosome. Chromatin, histone bound, which serves as a genome regulation point. No histones, uses supercoiling to pack genome. Proportion of non-repetitive DNA. The proportion of non-repetitive DNA is calculated by using the length of non-repetitive DNA divided by genome size. Protein coding genes and RNA coding genes are generally non-repetitive DNA. A bigger genome does not mean more genes, and the proportion of non-repetitive DNA decreases along with increasing genome size in higher eukaryotes. It had been found that the proportion of non-repetitive DNA can vary a lot between species. Some E. coli, Escherichia coli, as prokaryotes, only have non-repetitive DNA, while lower eukaryotes such as C. elegans and fruit fly still possess more non-repetitive DNA than repetitive DNA. Then, when it comes to higher organisms, higher eukaryotes tend to have more repetitive DNA than non-repetitive DNA. So in some plants and amphibians, the proportion of non-repetitive DNA is more than 20%, becoming a minority component. Then the proportion of repetitive DNA is calculated by using the length of repetitive DNA divided by the genome size. So there are two categories of repetitive DNA in genome: tandem repeats and interspecies repeats. These are the two types of categories we use in calculating proportions of repetitive DNA. So tandem repeats are usually caused by spillage during replication, unequal crossing over, and gene conversion. Satellite DNA and microsatellites are forms of tandem repeats in the genome. Although tandem repeats count for a significant proportion in genome, the largest proportion in mammalian is the other type, interspersed repeats. Interspersed repeats, because there's no way one person can replicate himself. Every child that you bear looks exactly like you with the same features, because there's an interspersion. The mother will bring some amount of genes, the father will also bring some amount of chromosome. They come together and form an entire human being. It does. Humans do not undergo asexual reproduction. We undergo sexual reproduction. That is why we have the interspersed repeats, unlike in certain organisms and plants. Interspersed repeats mainly come from transposable elements, but they also include some protein coding gene families and pseudogenes. Transposable elements are able to integrate into the genome at another site within the cell. It is also believed that transposable elements are an important driving force on genome evolution of higher eukaryotes. They can be classified into two categories: class 1, retransposons, and class 2, DNA transposons. So retransposons, that's class 1. They can be transcribed into RNA, ribonucleic acids, which are then duplicated at another site into the genome. Retrotransposons can be divided into long terminal repeats, LTRs, and non-long terminal repeats, non-LTR. So the long terminal repeats are similar to retroviruses, which have both GAG and polygenes to make cDNA from RNA and proteins to insert into genomes. But the LTRs, that's the long terminal repeats, can only act within the cell as they lack the EMV gene in retroviruses, the environmental gene in retroviruses. It has been reported that LTRs consist of the largest fraction in most plant genome and might account for the huge variation in genome size. Now, the non-long terminal repeats, non-LTRs, can be divided into long interspersed elements, short interspersed elements, and Penelope-like elements. Long interspersed elements lines are able to encode two open reading schemes to generate transcriptase and endonuclease, which are essential in retransposition. So the human genome has around 500,000 long interspersed elements, taking around 17% of the genome. Short interspersed elements are usually less than 500 base pairs and need to co-opt with the long interspersed elements machinery to function as non-autonomous retrotransposons. So the allele element is the most common sign found in primates, and it has a length of about 350 base pairs, and this takes about 11% of the human genome, with around 1,500,000 copies. So you realize that when it comes to genome, we tend to deal with huge numbers. That is why from 1995 till now, they are still in the lab, in alio and in silico, in vivo and in silico. We are still researching into it. We want to see how God created man, everything that is related to man, and we are still researching. We've not found out yet. So you see how nature is wonderful. DNA transposons. DNA transposons generally move by cut and paste in the genome, due to the act of transcription or transcriptase. When the double helix stranded structure unwinds, so DNA transposons generally move by cut and paste in the genome, but duplication has also been observed. Class 2, the transposing elements do not use RNA as intermediate and are popular in bacteria. In metazoan, it has also been found. Genome evolution. Genome evolution. So genomes are more than the sum of an organism's genes and have traits that may be measured and studied without reference to the details of any particular genes and their products. Researchers compare traits such as karyotype, that's the chromosome number, genome size, genome order, codon usage bias, and GC content, guanine cytosine content, to determine what mechanisms would have produced the great variety of genomes that exist today. Duplications play a major role in shaping the genome. Duplication may range from extension of short tandem repeats to duplication of a cluster of genes, and all the way to duplication of entire chromosomes or even entire genomes. Such duplications are probably fundamental to the creation of genetic novelty. Horizontal gene transfer is invoked to explain how there is often an extreme similarity between small portions of the genomes of two organisms that are otherwise very distantly related. Horizontal gene transfer seems to be common among many microbes. Also, eukaryotic cells seem to have experienced a transfer of some genetic material from their chloroplast and mitochondrial genomes to their nuclear chromosomes. Recent empirical data suggest an important role of viruses and subviral RNA networks to represent a main driving role to generate genetic novelty and natural genome editing. Now, in review, a gene could be described as the basic unit of genetic information present on DNA, the chromosomes and chromatin of an organism. Structure of a gene consists of many elements, of which the actual protein coding sequence is often only a small part known as the editors. A gene may also include DNA regions that are not transcribed, as well as untranslated regions of the RNA. These non-coding regions are called introns. A gene requires a promoter sequence or region in order to initiate transcription. The promoter is recognized, bound by transcription factors and RNA polymerase. The recognition typically occurs as a consensus sequence like the Tata box. A gene can move or have more than one promoter, resulting in messenger RNAs that differ in how far they extend in the 5' end. Eukaryotic promoter regions are much more complex and difficult to identify than prokaryotic promoters. And generally, genomes differ from prokaryotic genomes as outlined. Genomes refers to all DNA contained in an organism or a cell, involving or including the chromosomes as well as the DNA in mitochondria. Then prokaryotes gene expression typically regulated by an operon, the collection of controlling sites adjacent to polycistronic protein coding sequences. Eukaryotic genes also are regulated in units of protein coding sequences and adjacent controlling sites, but operons are not known to occur. Eukaryotic gene regulation is more complex because eukaryotes possess a nucleus. Transcription and translation are not coupled. Two categories of eukaryotic gene regulation exist. Short-term genes are quickly turned on or off in response to the environment and demands of the cell. Long-term genes are for development and differentiation. So that ends Unit 2, Session 5. We are on the final lap for today, and that is going to be Unit 2, Session 6, the flow of genetic information. The flow of genetic information. So at the end of this session, we should be able to explain the flow of genetic information. Then again, explain the central dogma theory of molecular biology. So what is central dogma theory of molecular biology? It was proposed by Francis Crick in 1958 to describe the flow of information in a cell. It explains how information stored in DNA is transferred residue by residue, bit by bit, by RNA, which in turn transfers the information residue by residue to protein. So from messenger RNA, move on to ribosomal RNA, then to transfer RNA, right? So the primary repository of genetic information is typically DNA. DNA is the hub, and it can be RNA in some organisms, which must be replicated with fidelity and transmitted to subsequent generations during reproduction to propagate the species. So DNA is copied or transcribed into RNA. Note, DNA is copied or transcribed into RNA through a process known as transcription. So it is transcribed or copied through the process known as transcription. We would in turn look at translation. So the resulting transcript, if protein encoding is subsequently decoded into protein by translation, thus RNA or messenger ribonucleic acid acts as the intermediary from the DNA. The intermediary is messenger RNA, intermediary information that can be translated. Somebody speaking Spanish needs an interpreter to translate it into Akan. So the intermediary, the interpreter is the messenger. Is that right? So proteins in large part constitute the major constituents of cells. So the set of messenger RNAs expressed in a cell and their protein products largely determined the phenotype, outward characteristics, type of cell. So the structure of DNA as a double helix with base pairing facilitates the replication and transcription of the information contained therein. So what is the central dogma of molecular biology? The central dogma was proposed by Crick to help scientists think about molecular biology. It has undergone numerous revisions in the past 45 years. In the 1970s, a modification to the central dogma was made when it was observed that RNA can be converted to DNA with the help of an enzyme called reverse transcriptase. So in most organisms, this is a rare event, whereas for some viruses such as HIV, it is a necessary part of its life cycle. So you have DNA being converted to RNA, then the proteins come in. So inferior replication DNA duplicates. That's a single strands occurring. Then transcription. Transcription RNA synthesis. You have the nucleus, cytoplasm acting. Information that is being transferred from one state to the other. Then you have translation. You've spoken in Spanish. You have an interpreter interpreting it into Hausa, Gan, Ewe, or Akan. So schematic representation of the central dogma of molecular biology to illustrate the flow of genetic information, how information is flowed, is processed, is passed on from one cell to the other. So the central dogma explains how information stored in DNA and subsequently transferred.
Doc, can you go back to the diagram again and explain to me again?
So you have a double helix structure. The long and short of it is that how is information transferred? The double helix structure of the DNA has to undergo denaturing by unwinding. You have a zip trousers. You want to urinate. That is, you are passing on. You are passing on some urine into the environment. You have to unzip before you pass on the urine, or else you soil yourself. Is that clear? That is exactly what it takes place up there. DNA is converted, so it unwinds. The DNA has now unwinds, and you have the complementary base pairings in the nucleus. So one has to leave the nucleus into the cytoplasm, and that has to be the intermediary. That is the messenger RNA. At this point, it is leaving the nucleus into the cytoplasm. It doesn't make it a DNA again. but makes it what? A ribonucleic acid. Okay, and that is the information replication DNA, because it has to double itself, and in doubling, one has to leave for the other pair. So the information, you see the arrow that has to do with the DNA moving to RNA. So they move from the nucleus into the cytoplasm, and that is how information is transferred. So the information on the DNA strand is being carried by the messenger RNA into the cytoplasm, which also releases the information to ribosomal RNA. And the ribosomal RNA are found on protein, on some amount of proteins that are hovering in the cytoplasm. Then the information will be carried from the ribosomal to the transfer RNA, because they transfer the information. That is why you have a nucleus, a line which is green, okay? And beneath it, you have the cytoplasm. So you see information is traveling from the nucleus into the cytoplasm. And that is when translation occurs. And translation is where proteins are formed. Protein synthesis occurs. So that informs the diagram. So the central dogma. Is somebody calling? Can I continue?
Yes, Doc.
All right. So the central dogma that explains how information stored in DNA and subsequently transferred residue by residue, bit by bit, to RNA, which in turn transfers the information residue by residue into protein. That is what the central dogma is about. Information is transferred from one stage to the other. So the primary repository of genetic information is typically DNA. DNA is the hub. Then it further divides into what? RNA. But can be RNA in some organisms, which must be replicated with fidelity and transmitted to subsequent generations during reproduction to propagate the species. So the DNA is copied or transcribed into RNA through a process known as transcription. The resulting transcript, if protein encoding, is subsequently decoded into protein by translation. Hence, the RNA or messenger RNA acts as intermediary information that can be translated into protein. Proteins, in large part, constitute the major constituents of cells. The set of messenger RNAs expressed in a cell and their protein products largely determined the phenotype of a cell. So we bring the lecture to a close this morning. So I'll make sure the slides get to you by your course rep so that he will distribute it to you. I believe he has made available the first lecture slide to you.
Yes, please.
All right. So,
Doc.
Yes.
Earlier, I was asking if we could get some questions that will be guiding us in our studies.
I am a very busy person, and this is the first time I'm handling this course, and so I can't go to such an extent. You are supposed to be doing that for yourself, okay? You come to the repository. You can do that there. So that has to do with that. So can your rep confirm that he has received the document? So Pokwajiman,
Sir, please, we have received the first light.
please, what about the second one? I'm sending it.
All
Okay.
right, sir. Thank you.
All right. So on that note, we bring our lecture to a close.
All right, Doc. Thank
Thank
So
you, Doc.
next week, God willing, there are some sample questions in the module. I will just put them to you so that you can keep trying your hand on them.
you, sir.
Okay, Doc. Thank
Thank
you.
you, sir.
Thank you, sir.
You're most welcome. Okay. So have a blessed weekend.
Same to you.
See you.
Bye.
Bye.
Hmm.
Yeah. Eh.
Johnny.
Hmm.
You can it.
So yeah,
Oh, good. Yeah. Really good. It is a good color. It is a really you will like have you here. Oh, very good. Yeah, very good. Very good. Very good. But I say good never reaching. Very good. Not take China. Very good. That one. Bye. That's how you know the couple never budget. I've been with one sicker with Billy Bua. About 5,000 feet. 1,000.
Let's go.